Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Researchers at the Stowers Institute for Medical Research, in collaboration with colleagues at Stanford University and Technical University of Munich, have developed advanced explainable artificial intelligence (AI) in a technical tour de force to decipher regulatory instructions encoded. Neural networks are powerful AI models that can learn complex patterns from diverse types of data such as images, speech signals, or text to predict associated properties with impressive high accuracy. However, many see these models as uninterpretable since the learned predictive patterns are hard to extract from the model. DNA bases, commonly represented by letters A, C, G, and T, encode not only the instructions for how to build proteins, but also when and where to make these proteins in an organism. The regulatory code is read by proteins called transcription factors that bind to short stretches of DNA called motifs. However, how particular combinations and arrangements of motifs specify regulatory activity is an extremely complex problem that has been hard to pin down. Now, an interdisciplinary team of biologists and computational researchers led by Stowers investigator Julia Zeidlinger, Ph.D., and Anshul Kandaje, Ph.D., from Stanford University, have designed a neural network, named BPNet for Base Pair Network, that can be interpreted to reveal regulatory code. This increased resolution allowed them to develop new interpretation tools to extract the key elemental sequence patterns such as transcription factor binding motifs and the combinatorial rules by which motifs function together as a regulatory code. This was extremely satisfying, says Zeidlinger, as the results fit beautifully with existing experimental results, and also revealed novel insights that surprised us. For example, the neural network models enabled the researchers to discover a striking rule that governs binding of the well street. Discovering that Nanog has such a pattern, and seeing additional details of its interactions, was surprising because we did not specifically search for this pattern. This is the key advantage of using neural networks for this task, says Iga Avsek, Ph.D., first author of the paper. For instance, the neural network first detects edges in the pixels, then learns how edges form facial elements like. Instead of learning from pixels, BPNet learns from the raw DNA sequence and learns to detect sequence motifs and eventually the higher-order rules by which the elements predict the base resolution binding data. Once the model is trained to be highly accurate, the learned patterns are extracted with interpretation tools. The output signal is traced back to the input sequences to reveal sequence motifs. The final step is to use the model as an oracle and systematically query it with specific DNA sequence designs, similar to what one. Since the approach is flexible and applicable to a variety of different data types and cell types, it promises to lead to a rapidly growing understanding of the regulatory code and how genetic variation impacts gene regulation. To enable other scientists to use BPNet and adapt it for their own needs, the researchers have made the entire software. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.